right. Well, thank you everyone for being here. It's a really amazing opportunity to give the first lecture of camp. In addition to being a humble country paleontologist, I also now work for this organization. So my life has taken a lot of turns, and I now am spending more time in the nonprofit world than I am in the science world. But I'm really excited to walk you through some of the science that I've done to get to this point and sort of introduce you to this really wonderful creature that I've spent a lot of my life studying, but also some interesting stuff about West Virginia that you may not know, and some a theme as suggested by Michael Stark, some philosophy behind sloths maybe we can apply to our experience here at camp. So this lecture is titled The Wild and Wonderful Sloths of West Virginia. If you've never been to West Virginia before, wild and wonderful is what we say about the state a lot in our self-aggrandizing. Does anyone here in the room, I'm gonna try to make this as interactive as I can. Does anyone here know which animal on this first slide is the sloth? Have a hand up, yes. The big one. So this giant creature right here is Megalonyx jeffersoni. That's means great claw, Jefferson's great claw. And so this scene probably depicts somewhere in late Pleistocene, Alaska. This animal harassing it is known as an entelodont. We sometimes call them a hell pig. It was a carnivorous pig-like animal that used to run around and is bothering these megalonyxes, and we're going to get into a lot more about what makes these animals so special and worth our study. So why study sloths? This is a question I get asked a fair bit because most people don't think that much about sloths. What they know about sloths are kind of the cute memes they see on the internet. But in reality, sloths have a lot of ecological importance, especially in the Americas, which is where they're from. So again, the largest animal in this image here is a ground sloth, Rematherium. This is, we've shifted, we're probably around the same time in the late Pleistocene, but we're now in what might be like Florida today. And what's really interesting about this painting is that we have in the foreground a tapir with a little baby tapir and some paleolama camelid-like animals. And in vertebrate paleontology, there's a lot of focus on these kinds of animals. They're really diverse, they have a lot of modern relatives, they produce a lot of fossils, but if you only focus on those animals that are easy to study, easy to get your hands on, easy to answer questions about, and you ignore the animal the size of an elephant, then I think you're probably missing a fair bit about that environment and how it actually functioned as a functional ecosystem that doesn't really exist today. So it's kind of an interesting relic of the past that we don't have a lot of modern examples of. So we are here at the National Youth Science Camp, just outside Bartow, West Virginia. If we drove about 2 hours south to Monroe County, West Virginia, it's an area that is rife with caves. You'll notice that Oregon Cave is the name of one of the town. Another town's called the Sinks of something, mines, springs. So it's an area that has a lot of fossils. And this endpoint on our route here is actually the cave where the first ever ground sloth fossil was discovered, ever. So it's here in West Virginia. It's called Haynes Cave. When you arrive in Monroe County, it's a lot of rolling hills, beautiful farmland. It's a nice spring day when I was there, and if you know where to go, it's not like a super publicly accessible cave, but if you know where to go, you'll find this hole in the ground. And this hole in the ground is the entrance to Haynes Cave, where the first ground sloth fossil was ever found. Now you'll see there's these weird grates on the front of the cave. Anybody know what those are for? Yes? Bats. It's a bat gate. So this grate is narrow enough that a human cannot fit through it, but it's easy for something that echolocates and flies to fly in between without hitting them. So it's a way for bats to get in and out of the cave without letting humans in and out of the cave, which is important because, yes. Disease, one disease in particular. Does anyone know the disease that is affecting bats here in North America? Okay, yes. White nose syndrome, or white nose fungus, yes. So it's a fungus that infects the bats. It actually wakes them up during their hibernation. And so if they can't successfully hibernate over the winter, they burn through too many of their calories and they essentially starve to death before they can make the spring. So really sad. We were trying to prevent the spread of it. I'm sure the people who go caving might learn a little bit more about that. But it prevents people from getting in. It also prevents people from taking things out of the cave, leaving graffiti, things like that. So if you know the people who manage this cave, which is the West Virginia Cave Conservancy, they will tell you the access code. There's like a tiny little padlock that if you know where to reach, you can unlock one of those gates. You still have to like crawl through and get on the ground, but it is possible to get in. So this is not a photo from the inside of Haynes Cave. This is just a generic photo that I found. But One thing I've learned, I don't have a ton of caving experience. I was talking with some of the caving coordinators about that. So the times I've been in Haynes Cave, none of my photos look very good because I don't have the right lights or setup or anything. So generic cave, but we can imagine that this is Haynes Cave sometime in the late 1700s, and people were in caves in this part of the country a lot in the late 1700s. Does anyone know why? There was an important economic resource that you could get from caves. Nope, not gold or silver, not coal, not iron, not copper, not quartz. Bat poop. Yes. So bat poop, guano, when it falls on the cave floor of these limestone caves, it has a chemical reaction with the caves that performs a potassium nitrate salt called saltpeter, colloquially. And saltpeter is really important in the manufacture of what? Gunpowder. So we had just fought a war for independence. And the War of 1812 hadn't happened yet, but I think we were smart to start stockpiling ammo. So to make gunpowder, you need saltpeter. So you have these guys in the caves mining saltpeter. The way you mine it is pretty gross. You like shovel up all this bat guano that's accumulated over decades, if not centuries, and you sluice it with water and separate out the minerals. But this is also West Virginia, so I have to imagine the guys were also making a little moonshine on the side. And I always think it's so hilarious. And it's called moonshine because you would do it at night to avoid the authorities. But another thing a lot of early colonialists did is they would do it inside caves because it's much harder. You can't see the flames from the still. But it also means that you are distilling a flammable product while making a precursor to gunpowder in a closed cavern. Doesn't seem like the smartest move, but that's how it went back in the day. So there are these saltpeter miners. We don't know their names. Their names have been lost to history, unfortunately. But they find this thing they can't really understand. They find these couple of bones, and they can tell, they know enough to know that these look like they're from the hand of some animal. So they give the bones to the guy who owned the land. And the guy who owned the land knew somebody who lived relatively nearby who would be really interested in learning more about these bones, and he went ahead and sent them to this guy. And the guy he sent it to lived here. Does anyone know where this is? Monticello. Yep, so who lived in Monticello? Jefferson, problematic fave Jefferson. He actually was the first author to describe a sloth species in the scientific literature. He did it as he was becoming vice president of the United States, so kind of an interesting confluence of science and politics. And the paper he wrote was published so the way scientific papers used to be published is they would get read at a conference. So the reason that you would have these scientific conferences is because printing was very expensive, shipping was very expensive. So instead you would get all the scientists together in a room, just like we're doing, and then Jefferson would have written his manuscript, and somebody, maybe Jefferson, maybe somebody else, would get up and literally read it to the audience, and that's how that knowledge would get spread. And then later it would get published as an actual written piece. So it got read at a meeting of the Philosophical Society of Philadelphia in 1797, and then published in print in 1799. And the title, for those of you who can't read the old-timey script, is A Memoir on the Discovery of Certain Bones of a Quadruped of the Clawed Kind in the Western Parts of Virginia. So you have to remember this is the late 1700s. This was still where we are now was still part of Virginia. Virginia was a much larger state. It didn't separate. West Virginia didn't become its own state until 1863. And Jefferson took a look at these bones and concluded that it was probably some kind of lion. And that's why he named it Great Claw. Megalonyx means great claw. Because all he had was the claw to go on. And Jefferson was also in sort of a scientific feud with this guy in France named Buffon, who was another naturalist. And Buffon had this theory that American animals were worse than European animals. And if you think there's probably some racial undertones there, you're right. So Jefferson was very keen to prove that American animals were just as good, if not better, than European animals, up into the point of sending Buffon an entire moose to be like, yeah, look at that. So, so Jefferson, loved the idea of having this giant lion-like animal in America and not in Europe. And when he was getting ready to send Lewis and Clark into the interior, he actually showed them these sloth bones and things like mammoth teeth because, again, this is 60 plus years before Origin of the Species is published. Extinction isn't really a concept in science yet. Like, it wasn't really understood that sometimes things stopped being around forever. So he kind of warned Lewis and Clark and said, yeah, I think there's a giant animal with claws out there. Be careful when you head into the interior of the United States, because who knows? So what we now know today is what he had found was a sloth. And sloths today used to be described as a group called edentata, which means without teeth, because some of the sloth's closest relatives, like anteaters, don't have any teeth. They used to include aardvarks and pangolins in this same group, edentata, but as we've gotten better at doing genetic analysis and found more fossil evidence of the different interrelationships between these animals, we realized that edentata is not really a thing, and we renamed the group Xenarthra, and it just contains sloths, armadillos, and anteaters. Now, Xenarthra refers to the vertebral column of these animals. They have strangely jointed. Xenarthra means strange joint. They have a very strange articulation to their vertebral column. Does anyone know why that might be especially beneficial to this particular group of animals? Say it louder, I heard it.
So they can roll up.
Bingo. Armadillos can roll up. So you have to have a pretty flexible spine to be able to roll up, yeah. Yeah, it does land on its head. So yes, so exactly correct. It's so they can roll up. And if you've ever heard the thing about sloths being able to turn their head like 270 degrees, that's part of, again, these strange vertebral columns. So, you know, now that we've dispelled the myth that aardvarks and pangolins get to be a part of this group, we now know that we have our family photo here of the Xenarthrans, and it includes basically two main branches. One branch is called Cingulata. That includes all the armadillos and all the extinct armadillo-like things like pangolins, not pangolins, things like glyptodonts and pampathirs. Glyptodonts are those big, almost Volkswagen Beetle-sized animals with like the spiky tail. They look almost dinosaur-esque, but they're actually related to armadillos. And then anteaters are their own branch within the other group called Pilosa. And then we have Folivera, which means leaf eater, which includes the two types of sloths we have alive today and all extinct ground sloths. So let's get into that a little bit. Sloths today there's two genera. One is the Bradipus genera. You might have heard sloths referred to as three-fingered and three-toed and two-toed. The confusing thing about that is the difference in digits is actually on the front paw or hand. So it's really, there's a two-fingered sloth and a three-fingered sloth. I might mix up and still call them toed instead of fingered, but it's one of the things we're trying in the sort of community to just be more clear about the difference between these two animals. So we have 4 kinds of three-fingered sloth. One of them is the recently described pygmy three-fingered sloth, so that's relatively new. It used to be really convenient because there used to be 3 three-fingered sloths and 2 two-fingered sloths, and then we had to go and find another one and make it complicated. You can get a sense of scale with my face right there, and yeah, they do look like they have a perma-smile. His claws are on top of my head because he's testing if I'm a good enough branch for him to climb on and get away from me. So that's what's happening there. And then we have 2 species of our two-fingered sloths. I'm not going to get into specifics between different species. Just there's in general 2 different types of sloth alive today. Go ahead. Okay. So this list here, I don't expect anyone to be able to read it, but this list here is all the different genera that have been described in the fossil record. So it's about 90 genera. So we have 2 genera alive today. We used to have close to 100. There's a thing in paleontology called lumping versus splitting. If you're a researcher and you're trying to get a paper out, it benefits you if you're describing a new species. So this might be an overrepresentation of the diversity a little bit. It probably could stand for some lumping of things together. You can imagine it's hard in paleontology to tell, did I just find a juvenile and describe it as a new species? Did I actually just find a male, female, but they're different enough that they look like different species? So those are the kinds of questions we ask when we're figuring out these taxonomies. But you can see the range in sizes. So we have a roughly 2-meter tall person here for scale. And then we have one of the smaller ground sloths, the Appalachia, which is still probably twice the size of a modern sloth, up to some of the elephant-sized sloths like Eremotherium and Megatherium. And if you see these animals depicted on their hind legs and think, really, they were on their hind legs? Yes, they were definitely up on their hind legs for part of their life. We have trackways of these 2 giant animals, and it's only the feet. There's no front limbs touching the ground. Their tail is kind of used as a tripod. So kind of cool. And then this here is a chart of mitogenomic data, or which is it mitogenomic, or it's proteomics data. So this is data that looks at the genes that encode for different proteins, and you can use that to figure out how long it's been since 2 animals shared a single common ancestor, so you can get a sense of the time it's been since they diverged from one another. So we have here a bunch of different xenarthrans, including in the red boxes are 2 living xenarthrans. And when you look at the point where those 2 lineages diverged, it's about 26 million years ago. And these animals have a lot in common, but from everything we can tell, they evolved from 2 separate lineages of ground sloths and haven't actually shared a common ancestor for 26 million years. The dinosaurs went extinct 65 million, so that's almost halfway back to when the dinosaurs went extinct. These 2 lineages diverged from one another and haven't really been that closely related since, which is just wild to think about.
Oh, so this is similar to cladistics?
Yes, this is a one method of sort of clarifying cladistic relationships, yeah. So this is really weird. This is a theme I'm going to come back to over and over again with sloths. Sloths are weird. If you described a sloth to a scientist who didn't know anything about them, a biologist who didn't know anything about them, they might not classify them as a mammal. They might be like, wow, that sounds like a weird climbing reptile that grew fur. But nope, they are mammals. And we're going to talk a little bit more about the ecological importance. Yes?
Is it sort of like how like crabs have evolved separately multiple times, but like since they were common ancestor, were they like different enough, not common ancestor, that they sort of like evolved similarly since then?
So you're talking about, so the question for those who maybe couldn't hear was about the idea that crab-like forms have evolved over and over again. Carcinization is the term. And it's not quite that, because they are closely related. It's more, this is more an example of extreme convergent evolution. So you essentially had animals living in an environment, and these 2 different lineages of sloths, as they were transitioning from the ground to the trees, had a similar set of problems they had to solve. And evolutionarily, they both kind of went down the same pathway to solve those problems. So how do I climb? How do I get enough food? How do I stay warm? So they developed a very similar set of adaptations. So they superficially seem very similar today because they were solving similar problems using similar means. So we have here the origin of sloths. So the earliest sloth fossils we have are from southern South America and the sort of the Tierra del Fuego region. And this is the early Oligocene, so about 36 million years ago is when we start seeing the first xenarthrans, and then a little later than that we start seeing the first things we call sloths. One really important thing to note about this map, what's not on this map? Does anyone see an omission in what our modern continental structure looks like? Just focus on the Americas. Don't worry about anywhere else right now. Yeah, so I've heard a couple people say, there's no Panama, right? The continents are not connected right now. There were, it was warmer then, there weren't glaciers. This might look like an unfortunate preview of what's to come. But Panama had not emerged in between North and South America, so Central America is not really connecting these 2 continents, and South America hadn't been connected to another continent for a really long time. So it had sort of been evolving on its own path in what some paleontologists call splendid isolation. But what's really crazy about sloths, and this gets back to the idea of sloths are very sensitive, very persnickety animals, but in this instance, they went way outside their comfort zone because, well, before I even reveal. All right, who here knows that there are sloth fossils from their state or their home country? Yes, where are you from? Ecuador. Definitely sloths from Ecuador. All right, can you put your hand up? We're gonna try an experiment here. And we know Argentina had definitely, so all the Argentinian delegates, can we get those hands up? Because we know we have, this is basically modern day Argentina. What about, do we have any delegates? Where are you from? Oregon, we do have sloth, John Day fossil beds. Panama, we have, Argentina, yep. Chile, definitely sloth fossils from Chile. What about more US folks? Anyone else in the US? Yeah. Alaska, yes. Who would have guessed Alaska? So I mean, you've kind of given the big reveal. Sloths made it across an ocean, an open sea, probably a shallow sea, and they might have island hopped, but they crossed over in North America before Panama finished forming. There's some debate on when Panama finished forming, but sloths were definitely in North America before it finished forming. By the late Pleistocene, they are everywhere. So do we have anyone from New York? New York State? Yep, sloths from New York State. Do we have anyone from Florida? Sloths from Florida. Do we have anyone from Texas? Sloths from Texas. Arizona, New Mexico, Nevada, California, Colorado. Colorado, on top of a mountain in Colorado at like over 8,000 feet or something like that. So sloths went everywhere, including the Caribbean. I have a little island, or a little a little arrow going out into the Caribbean. So sloths spread far and wide up until the late Pleistocene. And does anyone know what happens at the end of the Pleistocene? Extinction. So when you start learning about geology, we have all these different time periods that have names like the Cretaceous and the Jurassic and the Triassic and all that. A lot of times those transition between periods are extinction events. Maybe not huge ones, but enough that it's noticeable in the fossil record. So the late Pleistocene extinction happens, and it's not quite a worldwide extinction, but it's essentially the end of the last ice age. Humans are arriving in North America. Something destabilizes the ecosystem such that large-bodied animals all go extinct. It's an extinction that seems to target large-bodied animals. There's some different theories why. We can talk about that more in depth afterwards if folks want. But I think it's important to, again, re-emphasize that as sloths are expanding all over the place, they're diversifying. They're finding different habitats to live in, different niches, different environments. And one way we estimate that in paleontology is looking at snout width. So this is probably intuitive, but you can imagine that an animal with a wider snout, wider muzzle, tends to just eat whatever. They tend to be not picky. They just kind of scoop up on the environment whatever they can get their face into. Whereas, so think of like a warthog, right, or feral pig, versus very slim-snouted animals tend to be more selective. They tend to be pickier. So you think like a deer, something like that. So sloths have one of the widest ranges of muzzle widths in the animal kingdom, which suggests a big diversity of habitats and dietary preferences. Again, just showing the differences in sizes that we're talking about here. And I argue that they were probably an important part of the ecosystem. So an animal that large, an animal as big as the tree, if it knocks over that tree, it's changing the environment. And sometimes those changes persist for decades, if not centuries, if not millennia. So I think they were probably similar in their importance to elephants in Africa today. And just to sort of demonstrate what elephants in Africa today are capable of, I have a short video. I'm glad the sound is working because the sound is about to get very intense. Yeah. That's a good question. I'll speak to that here in a second. So the title of that video when I saw it online was "Road is now closed." Also, like, it kind of makes the guys flying helicopters to cut trees down seem a little silly. Like, you could just get an elephant to come through. There was a question: why is this animal doing that? My theory, I think that's just a young bull elephant, and I think he just had a lot of testosterone and was just like, man, screw this tree. But it's also it gets back to this theme of just like getting outside your comfort zone. Like if something is like in the way of your comfort zone, you can just knock it over, put it in the road, make it the tourist problem. All right, so let's get down to some actual science. So I've given you guys a lot of background on sloths because I think it's really important when you delve into a new scientific topic that you actually understand the background of what it is you're delving into. Otherwise, the questions you're asking and the answers you're getting might not make sense without that context. So if sloths are so interesting and important, why haven't they been studied more? Why is there still so much work to be done? There's kind of a lot of reasons, but first I want to clear up a quick misconception that I've realized I need to start including in this lecture, and this may not be the exact right spot for it, but it's something I get asked, and so I want to clarify. What is a fossil? Does anyone have a good working definition for a fossil? Yes?
It's like a bone preserved inside of like stone or something.
Yes, a bone, but it doesn't have to be a bone. It could be, yes. A footprint, yes. Any ooh, you caught yourself. Nailed it. Any preserved remains, impression, trace. So you guys were all hitting on different parts of it. But any preserved remains of life, of a once living thing from the past. Now, in practical terms, we usually cut that off at about 10,000 years. So the late Pleistocene extinction is about when paleontology starts. Anything more recent than that is still sort of considered biology, and especially if you're studying like apes and humans, it's more anthropology. but it's just anything preserved that gives us evidence that life was once there. So it's a pretty broad topic. There's two major categories of fossils, body fossils and trace fossils. So body fossils are any aspect that tells you about the morphology of the organism that you're studying. So an impression of a leaf. I guess I can pull up my examples. So like T-Rex skull, very obvious body fossil. It's telling us about the skull of this animal that once used to live. An impression of an insect. This is a wasp that died in a lake and settled down to the bottom of a lake and got covered with very soft sediment. This is from, anyone know where this is from? It's from Colorado. It's from Florissant Fossil Beds National Monument. It is a bee. It is a bee. Or a Vespa. Not the scooter, but the actual animal. And this is a petrified log. So again, these are these are telling us about the morphology of these animals. An important misconception that I think a lot of people have is, you hear the terms permineralization, recrystallization. Obviously, this log has been recrystallized. It's had a mineral form inside of it that's made it much harder than it would have been naturally, and so it preserves into the modern time. But that doesn't have to be the case. A fossil can be the original material from the animal or organism that died. Original material still counts, it just has to be old. Trace fossils give you some evidence of a behavior. So something that happened caused by life that isn't necessarily informing you about the body of it, but it's telling you about a behavior. So somebody mentioned trackways, right? Trackways are a trace fossil. You can learn a little bit about morphology, but what you're also learning about is a behavior. How fast was this thing walking? What environment was it in? Footprint fossils are interesting because they form very quickly. Yes? If you think, so trace fossils are interesting because sometimes, I'm gonna answer that question. So this fossil here is a burrow from some worm that lived in the ocean back in the day. We've never found, in a lot of cases, you don't necessarily find the body of the animal with the trace fossil. And so there's a whole nomenclature of named trace fossils that have a scientific name that are based on a burrow, not any actual animal that's ever been found. With something like trackways, if you find a trackway that you can reasonably place based on other fossils found in the region to a specific animal, then if you have an idea of how long the legs are, you can use some pretty basic trigonometry to figure out how quickly would it had to have stepped using legs the length we think it had to make the trackway that with tracks this distance apart. Does that make sense? Yeah. Yes? Mm-hmm. Yeah. You would have to probably ask a dendrochronologist that question. I don't know exactly that question, but in terms of just finding the fossil, it really depends on the fossil site. So some sites, there's just fossils littering the ground, and you just walk like this for long enough, and then you finally see something, you go, oh, there it is, and then you start digging at the spot where you find it. Other places are active quarries, so you're actively digging. Other places, like the fossils like this wasp fossil, it's these paper-thin layers of shale, and you have to like lift them off with almost a razor blade one by one and find things embedded in there. So it really depends on the type of fossil you're talking about. And then this other trace fossil here, does anybody know what this one is with the two people standing inside the circle? It might not be big enough to easily tell. Yes? Yeah, it's a sloth burrow. So a sloth dug out a burrow big enough for people to walk through. This is it's in Bolivia on the border of Brazil. And you can tell a sloth did it because there are claw marks on the wall. So that's what these marks on the wall are. So these giant animals were digging mega burrows. Yes?
How far back does the burrow go?
I don't know that it goes much deeper than where the person taking the picture of the people standing out there is, but big enough for a sloth and probably a family of sloths if they lived in familial groups. When do they lose their energy? It's a good question. We're going to get to that. There's one last trace fossil that I can talk about in more detail. It's not included in this lecture, but I have done a lot of work with it, and it's feces, coprolites. So sloths have a pretty amazing coprolite record. Some of these caves that these sloths that we find, there's strata, there's different layers. We can tell like, oh, this layer of feces is from 27,000 years ago. This layer of feces is from about 12,000 years ago. So they were like creating actual layers of feces in these caves sometimes. So, and you can learn a lot. You can learn a lot, yes.
For the burrow, is there any other way of identifying that it was a sloth burrow other than the claw marks?
Size. So, I mean, the current largest land animal in South America, anyone know? Tapir. It's a tapir. Is the largest land animal in South America right now. The size of that cave burrow is about the size of a mylodontid sloth, and there just wasn't anything else that big in South America at the time. You know, giant anteaters are pretty big today, but like they don't have, anteaters have a terrible fossil record. Also, who are sloths related to? Armadillos. Armadillos are pretty good diggers. So they clearly share some sort of lineage with a digging animal. So we think that the claws that the sloths now use for climbing might have early on in their evolution been used for digging. So it's a good question. Yeah.
So you mentioned that you don't know whether the giant sloths live in familiar groups. Is there a way that you can find that out about extinct species?
That's a great question. And that's a lot of times where new projects start is you'll have an idea of like, oh, that's interesting. How would I test it? There are a couple of cave sites that do have sloths from different age classes living in them. So we clearly have some adult sloths, some juvenile sloths, and maybe even some like yearling sloths. So if you assume that they all died at the same time, then that would represent a familial group. But how do you test it? You know, the dating methods often have error bars in the thousands of years. So if you're trying to pin down the specific, this one spring, all these 3 sloths were living there together, it's just tricky. And so you have to come up with clever ways of testing things, ways of making your argument, ways of telling the story of the fossils that you found. So, you know, it's part of that CSI detective work of being a paleontologist, but it is also storytelling. You're also figuring out the narrative of what happened when this animal was alive, or these groups of animals. So that's a really good question. It's kind of the million-dollar question of paleontology. Mm-hmm. Yeah, so the question is, you know, with the strata of coprolites in the caves, what does that tell us about the periods of inhabitation? And what we think is going on there is modern sloths, as I'll talk about here in a second, are really bad at regulating their body temperature. They're not like us. They don't stay at 98.6 all the time. They go up and down. So if we assume that fossil sloths had similar problems with that process of thermoregulation, we can then think of them as using the caves as what's called a thermal refuge. Basically, caves tend to approximate whatever temperature is at the surface year-round. So it's the average surface temperature is the temperature inside the cave all the time. So if it's really hot out, the cave's gonna be a little cooler. If it's really cold out, the cave's gonna be a little warmer. And so if we can tie those, you know, if we date the poop and we know roughly when it came from, we can look at what the climate was doing then, and we tend to see that during periods of slightly cooler climates, sloths are spending more time in the cave. So they're probably just going there to stay warm, maybe a safe place to give birth to their young, hide from predators, things like that. But yes, we can use those different strata to learn about the different periods of when they were inhabiting it over generations. Yes. What were the predators of the sloth? Well, that's a great question too. Probably not humans, surprisingly. There's very little evidence of humans hunting sloths. Even after we arrived in the Americas, there's one site in New Mexico that has a sloth footprint and then a human footprint inside the sloth footprint. And as I mentioned, footprints only form within a couple hours, so these humans probably were stalking this sloth. But natural predators, it was probably things like saber-toothed cats. There was an American lion, and saber-toothed cats may have hunted in packs. So the thing about sloths is, in biology, you can grow so big that nothing will mess with you, right? That's kind of what elephants have done. It's kind of what other large-bodied animals do. You grow so large that nothing will mess with you, and then you just have to figure out a way to keep your young alive long enough that they can get that big, and then nothing will mess with them either. So there was an American lion that was much bigger than any cat alive today, bigger than Siberian tigers. There were American cheetahs that were about 50% bigger than African cheetahs. There's people who think that the saber-toothed cat may have hunted in packs. We know there were dire wolves that hunted in packs. So, you know, it was probably a variety of things, but there was also, you know, things like the California condor or the Argentinian condor. If you think about those as giant vultures, they needed giant carcasses to eat. So there's also probably they were just good food for scavengers on some level too. Yeah.
Like we know like the humans were like hunting giant animals. Is there something about this sloth that makes it not, we don't want it?
My personal theory. Well, what I know about sloths today is tree sloths, about 30% of their body weight is stomach. And so if you're not interested in eating a fermenting bag of leaves, you're probably not that interested. So 30% by weight, you're not getting a lot of meat on the bone. Modern sloths, again, we don't know for sure about fossil sloths, but modern sloths have almost no subcutaneous fat. So it would be very lean meat. Humans, we like fatty meats. We like bacon more than we like beef jerky. So, you know, it's, I just think they didn't taste that good, maybe. And I also think that like in hunter-gatherer societies, The evidence is pretty straightforward that the gathering that was mostly done by women provides way more calories than the men going off and pretending to hunt things and not doing a very good job. So just saying. And I think honestly, if you imagine these societies of like, it's mostly young men going out to hunt, looking for glory. I just don't think there was that much glory in being like, you killed the slow thing that can't run away. Like, get me a bison and we'll talk. So that's just me. I don't know that that's, and you know, we might still find evidence of people having eaten sloths. There's one fossil site that claims that they have evidence of tool marks. That's usually how we can see tool marks from humans look different than teeth marks from a dire wolf. So there's a site that claims they have evidence. I'm not super convinced by it yet, but we might find new fossils that show us something different tomorrow. Yes? Okay. Yeah, I have a North American bias with the literature, so I probably should look into more of the South American literature and see if there's better studies down there. So a lot of times what we do in paleontology, if you want to study an extinct organism, you sort of ask yourself, well, what's alive today that might be a good model organism to look at so I can sort of start thinking about how this organism might have functioned when it was alive. So this is a tapir fossil. Tapir fossils look almost exactly like a modern tapir. You almost cannot tell the difference if you didn't know how old the bones were that you didn't have a modern tapir on your hands. This is a paleo llama, a llama-like camelid, also from the Americas that, again, today we have alpacas and other South American camel-like animals. And then this is a paramylodon, a large ground sloth, and we have just nothing. So we have modern tapirs, modern llamas. But for sloths, all we have are these tiny little things that live in trees. We don't have anything on the environment today that looks and functions like a ground sloth. So when we ask ourselves as paleontologists the question, should I study modern sloths to have a better understanding of ground sloths? The answer is, we don't really know. But you have to try, and so that's sort of what I did for my PhD research. Yes? That's sort of what I'm building towards with the research that I did. I don't have a definitive answer, but I think, you know, it's worth running the same analysis. Like, we have a ton of data of the type I'm about to get into for these large-bodied herbivores. So, you know, we can say, well, does it make more sense to use a tapir data on a ground sloth because it's a large-bodied quadrupedal herbivore, or does it make more sense to use the tree sloth data? The problem is there was no tree sloth data to answer that question with. So part of what I did, as I'm about to tell you, is generate that data so we can at least start deciding as a paleontological community, does it make sense to consider tree sloths as the analogs? But they're very difficult to study, tree sloths, that is. One of the reasons it's so difficult to study is they're just hard to find. So there's a sloth in this photo, I promise. This is not a trick. This is a photo I took in Costa Rica, staring up into the canopy. I often joke that I will probably, if I keep studying sloths, I will probably die from some sort of terciopelo or fertile ants bite because I'm walking through a snake-infested jungle like this, not looking down at all. But does anyone think they can see the sloth? I'll zoom into the area where the sloth is. If you think you see it, put your hand up. All right, you're right up here up front. Go tell us where you think the sloth is.
Is it right in the middle?
Go ahead, get up and point to it.
Is it that?
Nope. Yep. Yep, that's it. So, just to orient you, this is the head here. This is one arm, the other arm, this is the body and kind of the butt, and then the legs are above it, so you can't see the legs. And this is a sloth on the move. This is a sloth going from tree to tree. We're talking about an animal that spends up to 20 hours a day resting in one spot. Human vision, because we are somewhat predatory by nature, very attuned to movement, right? Your peripheral, our peripheral cells, your peripheral vision, the cells are really good at sensing movement, which is why you think you can see something, off to the side when you can't really resolve it, but you know you see movement. So when sloths don't move, that's a way to avoid predators like us, just by not moving. And it's a very effective strategy, especially when you're looking up into the trees. It's backlit with the sun. There's a lot of termite and ant mounds up in the canopy, so they kind of look, when sloths kind of huddle up and pull their limbs in, they look kind of just like a termite or ant mound. So they're just really, and sometimes they turn green. They have algae that grows on their fur. It's like nature's best camouflage. So they're just really hard to spot. And then, okay, so now we want to study this sloth. We want to get some data. We found it. Now what? You still might be waiting a while, right? This is an animal that only comes out of the trees once every 2 weeks. So either you're going up to get it, or you got to wait, and that makes for a pretty long research project to get funded. So that's a great question. The question was, does it come down for water? For a long time, it was thought that this is a three-fingered sloth, that three-fingered sloths did not drink any water. If you're an animal that lives up in the canopy, if you're an arboreal animal, an interesting thing to consider, in a rainforest, the canopy is basically a desert because all the water falls through. It doesn't stay up in the canopy. So if you're an animal that needs water, you do have to solve that problem even in a rainforest where it rains every single day. There are plants, epiphytic plants that grow on limbs of trees that will collect water, and there are like species of frog that lay their eggs in there. So it's possible that the sloths would drink from there, but it's never really been observed. So we think that sloths get the vast majority of their water, it's called preformed, from food. So they're eating leaves. The leaves are going to have some amount of water in them, so they're getting some water just for the food they're eating. Very, very recently, within the past couple of years, we've caught on camera and video sloths coming down and actually drinking water, particularly in mangrove forests, where the roots of the trees are going directly into the water. And it's one of these classic things in science. Native people have been telling us forever that sloths drink water. Of course they drink water. We see it all the time. But of course the Westerners don't believe it until we catch it on film. So that was actually one of the papers that I published in during my PhD research, was the first recorded evidence of this type of sloth dipping its face into water and actually drinking water to get some of its water. So it affects, a lot about the conservation and the husbandry of these animals in captivity. So it's really important to answer the question, does this thing need water to survive? So, yeah. So sloths, I like to say, if we go back to this photo, they essentially live in an all-you-can-eat buffet, right? Their food is leaves. There are very few other vertebrates that live in the tropics that mostly eat leaves. So there's basically two types of sloths, and then howler monkeys, and that's pretty much it. So there's a lot of ants and different insects that eat leaves, but amongst vertebrates, because plants don't want their leaves to be eaten, they put a lot of chemicals in the leaves to make them not taste good to larger animals. So like, is there anyone here who just can't stand the taste of cilantro? It's got that, yeah, the soapy taste. That's a bitter alkaloid compound that the cilantro leaves is putting in its leaves to be like, don't eat me, leave me alone, let me photosynthesize. So those are called secondary metabolites. So plants will put secondary metabolites in their leaves to prevent vertebrates and other critters from eating them. So sloths, like I said, their stomach is about 30% of their body weight by mass. So they keep their stomach full at all times, which is really easy to do when you're surrounded by your food source and there's no one else competing with you for it. So they eat as often, basically as soon as food moves from their stomach into their intestines, they're eating more food to keep that stomach continuously full, because again, they don't have any fat. So like if you or I get really sick and we can't eat for a little while, our body relies on our fat reserves to keep us alive because you can't keep any food in your stomach. Sloths don't have that luxury. If a sloth gets sick, it does not have fat reserves to pull from because fat would make it heavier and would make it tougher to climb in trees. So instead, they rely on just having a stomach constantly full, living in an all-you-can-eat buffet. Yeah.
So now that we're getting evidence that sloths are drinking water or things that are happening that like the indigenous people said are happening, is there more respect within the scientific community for the indigenous people's words, or like trying collaboration between them?
Yeah, so the question is about, is science doing a better job of respecting indigenous knowledge and working with indigenous people? I think in broad strokes, yes. That's definitely a trend I'm seeing in paleontology, where we're trying to get away from what's called helicopter science, where a bunch of white people helicopter into a remote location, find a bunch of fossils, and then leave. That's no bueno anymore. So now there's a much stronger emphasis on working with the local people, working with local researchers, working with local knowledge. I can't claim that it's perfect yet, but I think we've made important progress. For example, if you're a paleontologist who wants to do work in China, China will not give you a research permit unless you have a Chinese scientist on your team as part of the project. And I think steps like that are really important, because it's like, yeah, you have to include the people and the places. And so like I worked a project in Panama that was collecting fossils from the Panama Canal, and part of the arrangement that we had with the Panamanian government was some of those fossils might come back to the US to get studied for a bit, but their home is Panama. They're going back. So we're not taking these things out of the country and just keeping them. We're gonna make sure that they get their final home in Panama with the Panamanian people, and I think that's good. I think that's progress. So, yeah. Yeah, so why is it that sloths have such a unique metabolism? I'm going to talk about that a little bit, but the quick answer is leaves are not a great food source. There's not a lot of nutrients in here. So your options for, if you want to eat just leaves as your diet, your options are like be the size of a gorilla and just have, be able to eat enough food that you can actually get some nutrients out of that, or digest it very, very slowly so you're extracting as much nutrients out of it as possible. So it's the cold brew method of digestion. They're just sometimes literally because their body temperature is low. I saw another hand back there. Is it? Yeah. No, I think they'll go extinct. So the question is, will they start evolving fat reserves with deforestation? Deforestation is a huge problem in Central and South America. Forest interconnectivity is a huge problem, monocultures. So we can talk more about some of the conservation challenges that sloths face. I work with an organization called the Sloth Conservation Foundation. One of the things that they do is they put up sloth bridges. So basically where roads have now come and divided the forest, and it's really hard because you want these places to be allowed to develop economically and to make lives easier for the people that live there, but you have to make sure you're doing it in a way that preserves the environment. And so, literally just stringing a rope across roads at frequent enough intervals that sloths, possums, squirrels, monkeys, all these animals that need to get across the road without getting hit by cars have a way to do it. Like little things like that can make a huge difference in making lives easier for sloths so they can actually stay connected with their community, because sloths aren't super social, but you still have to find a mate every once in a while. So it's important to have the forest connected enough to do that. Yeah.
Does creating locations like those sort of create a predator activity too, like area of vulnerability?
So the question is, does creating like forest corridors and things like that create predator hotspots? There's only really two predators of sloths. Does anyone know what, or modern sloths anyway? Does anyone know what they are? Jaguar's one of them, and I imagine a large size. Other cats like a puma or something. But yeah, jaguar's a big one. No, it's not snakes. There are no snakes that'll mess with the sloth. Nope. People. Well, there's a huge problem in Central South America with feral dogs and sloths. So another reason to keep the forest, them in the canopy is when they have to come down to the ground. A lot of times feral dogs will attack them. But there's a natural predator of sloths that I'll reveal in a minute. I'll keep it. Yeah. Eagles, harpy eagles. The national bird of Panama, now extinct in the nation of Panama. Not great. Well, we almost did it to our eagle too. So it's, conservation challenges. Yes?
With what you're mentioning about Panama and like traveling, how are fossils transported like long distance?
How are fossils transported long distances? It depends on the fossil. So like when we're out in the field, it's usually just rolls of toilet paper for small stuff. Sometimes in the field, if you find something really, like if you find a T-Rex skull in the field, a lot of times, or a large dinosaur bone, sometimes, depending on the resources you have available to you, you can put it in plaster out in the field and then jackhammer the rock out underneath it, so you're removing all the rock with it so that it's protected by the rock that's protected it for millions of years already, and then you don't do the final prep work until you're back in the lab. So, in Jurassic Park when they're like brushing a little bit of sand off a perfectly preserved bone, never happens like that. You're finding a broken puzzle, a broken 3D puzzle that you then have to extract and put back together somewhere else, usually. And sometimes the blocks of rock that get jagged, like they're big enough that you have to call in a helicopter to like helicarry it out. There's other different fossil sites. So like, where's the Tennessee delegates? Yes, where are you from in Tennessee? East Tennessee. So East Tennessee State University, ETSU, the Gray fossil site, one of my favorite fossil sites in the world, is they were building a highway bypass and just kept finding rhino and tapir bones. The construction crews did. And so they did report it, because that's what you're supposed to do. A lot of these construction crews have a geologist or paleontologist who consults on these projects, that in case you find something, and an archaeologist in case you find human stuff, that if you find something, you're supposed to stop and make sure that it's not so scientifically valuable that you shouldn't put a road through it. And in this case, the Tennessee state government decided, yeah, we should not build this road. We should build a science center right here. And so it's like the most convenient fossil site I've ever been to, because your prep lab is like 100 yards away from where all the fossils are. So you just walk out the back door of your prep lab, pick up whatever tapir bone you found that day and walk it back inside, and you can get started. So it varies based on the fossil site, but yeah, it's one of the major challenges of paleontology for sure. Yeah.
I have a question, right? I just want to make sure I heard you correct. You said that there isn't much fossils of the sloths, aren't there?
There's no fossils of tree sloths. We have a lot of fossils of the ground sloths.
Right, but the tree sloths, is it because
That's probably part of it. So the question was, do we have any fossils of tree sloths? And the answer is no. We don't have a single fossil. We have a couple of species of sloths, ground sloths, that were small enough that they probably could have climbed into the trees, but they're from the Caribbean, so they're not the ancestors of the sloths we have today. There's this entire field of paleontology called taphonomy. That's literally the science of how does an organism go from being, like, the moment it dies to preserved all the way up until people finding it. And it's a huge field of study. You can imagine all the variables that come into play with that. And it is interesting because we think that forest canopies are environments that don't tend to fossilize very well because of exactly what you're saying. You know, they're not close to the ground. They're not getting covered with dirt right away. Rainforests are also incredibly bioactive environments, especially with the roots of the plants and stuff. So roots kind of tend to destroy fossils. So if you've got an area where there are a ton of plants and a thin soil horizon and things are constantly growing, there's a lot of energy in that system because it's the tropics. It's hot, there's sun, there's a lot of biotic life, there's insects, there's scavengers. So yeah, it's probably just not a very good environment for preserving fossils, especially animals up in the trees. So yeah, I think you're on the right track. Yeah.
I have a follow-up to that question then. Is there like a significant time gap that you now have between the ground sloths and the tree top sloths? And
So-
then would you be able to, I guess, classify or guess what kind of sloth were in between?
Yeah, so the question is there a time gap between ground sloths and tree sloths, and how would you clarify the animals, the sloths that were in between? That's a pretty active field of study in sloth paleontology right now, but from what we can kind of tell, it's almost every single ground sloth is probably in between the two tree sloths that we have. So we think that the bradypus sloths are almost an outlier, and then everything else is nested with the two-fingered sloths. And there's no gap, so as far as we know, there were tree sloths during the late Pleistocene extinction. They just survived, and the ground sloths just didn't survive. And the ground sloths on the continents of North and South America went extinct about 10, 11,000 years ago as the last ice age was ending. The Caribbean sloths actually survived until about 4,500 years ago, which is incredibly recent. Remember I said that the Great Pyramids in Egypt were finished by the time the last ground sloth went extinct, which is kind of crazy to think about. So, what else makes sloth weird? These are drawings of sloth teeth. So sloths, remember the group used to be called edentata, which means without teeth. So anteaters truly have no teeth whatsoever in their mouth. Armadillos and sloths do have teeth, but their teeth are different than pretty much any other mammal's teeth in that they don't have enamel. They've lost the ability to produce enamel. So enamel is the outermost layer of our teeth. It's the hardest substance your body produces. It's about a 95% hydroxyapatite matrix, so it's basically a rock that your mouth grows twice in your life because it's expensive. It's metabolically very expensive to grow a really hard tooth. So sloths probably evolved from insectivorous, so an insect-eating ancestor. If we think about anteaters and armadillos, it's sort of eating insects. And what we see in a lot of mammals is mammals that focus primarily on eating insects tend to lose their enamel. So aardvarks also don't have any enamel on their teeth. Pangolins also have completely lost their teeth. And so what happened with sloths is they did this reversal where they were going down this sort of insect-eating path and then went back to a completely herbivorous path, and leaves are much harder to orally process than insects. So now they've got a problem. They've got no enamel, so their teeth are physically softer because their teeth are just dentin, which is the inner layer of our teeth that still has like nerves and blood vessels and is still alive. The enamel on the outside of your mouth, it's done. It's not being produced anymore. It's just a rock inside your mouth for chewing your food. So the solution sloths came up with, evolutionarily speaking, is they have hypermineralized their dentin, so their dentin is harder than our dentin, but still softer than enamel, and their teeth grow continuously throughout their entire lives. So you think about like a rodent, the two front teeth of a rodent, the incisors, they grow continuously, so rodents have to keep gnawing on stuff to keep those teeth from getting too long. That's the sloth's entire mouth, but it's all dentin. So it's this softer material. Why is that interesting for the fossil record? Because hard materials preserve better in the fossil record. Soft materials don't preserve as well. So we again have this preservational bias where the tissue that we like to study in all other mammals doesn't exist in sloths. So, and then we've talked about temperature regulation. So here is a chart that has the temperature in Celsius on the y-axis and the time of day on the x-axis with N at noon and M at midnight. And this is the top curve here is an individual sloth body temperature, and the bottom curve is the ambient temperature of the outside, and you can see that the sloth's temperature is fluctuating with the environment, right? As the environment gets warmer, the sloth gets warmer. As the environment gets colder, the sloth gets colder. And we're seeing, 5+ Celsius shifts in temperature, like shifts that would put any one of us in the hospital. If our body temperature shifted that much, you would be close to death. And this is like a normal day for them. And I think it's so funny because if you look closely at this one part of the chart, this little cloud here is showing us that it rained. And when it rained, there's another dip. So like even just getting wet causes their temperature to dip even further. And they live in a rainforest. So this happens a lot. So that's really weird. That's like, again, if you saw that and didn't know this was a mammal, you'd think you were looking at like a turtle or something. And then we talked about the metabolism. Okay, so this is a chart that shows the basal rate of metabolism. So essentially your metabolism when you're at rest. And that's measured as a function of CO2 production. So literally you put an animal in a tank filled with oxygen, and you just measure how much CO2 is produced by that animal over a period of time, and that gives you sort of how fast are they breathing, therefore how fast are they processing everything internally to their body. And then on the x-axis is body weight. So it's been known for a long time in science that, especially in mammals, body weight scales inversely to metabolism. Put another way, bigger mammals have slower metabolisms. So think about the difference in a squirrel or like a spider monkey or a capuchin monkey versus a gorilla, right? Think about a rabbit versus an elephant, right? So the bigger you are, the slower you tend to be. And everything fits nicely on this trend line, except for the sloths. So the sloths fall way below the trend line. So again, if I gave you the basal metabolic rate of a sloth, an animal that weighs a couple of kilograms, you can easily hold one in your arms, and said, how big do you think this animal is? You would estimate that it's the size of like a tapir, the largest land animal in South America, but it's not. It's this tiny little animal that lives in the trees. So again, very strange. Sloths, we talked about a little bit already, digest their food very, very slowly. So this is 4 different sloths that were done a feeding study where to figure out how quickly food passes all the way through the animal, you give it food marked with a marker, some sort of chemical tracer that isn't going to harm the animal but is detectable in the feces that comes out the other side. So you give it to the sloth in a known amount, and then you just keep measuring the poop that's produced until you've recovered roughly 100% of the chemical. Make sense? So these are the proportion of the marker recovered. So you can see they all kind of arc towards 100%. And then on the x-axis is time. And we're looking at an average of almost 300 hours. That's 12 and a half days. By comparison, there's 24 hours there. Most mammals, us, horses, elephants, like really large bodied, it's all about 24 hours. It's sort of a mammalian standard, except for sloths. The next longest, does anybody know what the next longest or slowest digesting mammal is? Not elephants, they're related to elephants. Think of an elephant, but in the ocean. Not whales. Manatees. Sharks are not mammals. They don't nurse their young. It's manatees. And manatees, it's about 150 hours. So they're twice as long as the next longest digesting animal. So it's a really interesting and strange life history trait. So, okay, they're weird, but science should be about answering questions. What are the questions? Okay, this is where I'm going to get this is going to get weird. There's actually debate about ground sloths being strict herbivores versus maybe occasionally eating meat. That's from Futurama, if you've never seen it. Yeah, actual depiction. That's cave art. No, so there's this South American paleontologist who came up with this theory that maybe Megatherium, the largest ground sloth to ever evolve, was a secret meat eater or maybe a scavenger or an omnivore. And the reason he came up with this theory is there are these fossil sites in South America that produce tons and tons of sloths of different species and other herbivorous animals, and we haven't found any predators at these fossil sites. Now, in most ecosystems, you have more herbivores than predators just because metabolism is an inefficient process, so you need more biomass of herbivores to support a smaller biomass of predators. That's just, it's fundamental physics at a certain level. Energy transfers are inherently inefficient, so you're going to lose energy going up the food chain. You have to make sure you have enough biomass to support everything. So his solution to not finding any carnivores at this fossil site was to propose that maybe the ground sloths were eating the meat and there were just these cryptic scavengers, up to the point of suggesting that this sloth, remember those glyptodonts, those armadillo-sized Volkswagen Beetle-looking things, were running up and just flipping those on their back and like scooping out the insides with their giant claws. So a pretty far out there theory, but one of the things we've talked about today is, okay, so somebody proposes an idea like that, how do you test it? So there's one really cool technique, and it's called stable isotope analysis. So hopefully this is review for all of you, but an isotope is an atom that has an extra neutron or two or sometimes more. So it's a heavier version of the atom than we usually see. So the idea behind stable isotope analysis is you are what you eat down to the atomic level. So those of you who had cereal grains, like in cereal this morning versus those you had oatmeal, you had like a slightly different cocktail of atoms that you were ingesting, and those literal atoms are going to become your skin, your hair, the blood, the tissues that reproduce over time. And if you know what that blend going in is, then you can kind of measure what that animal might have been eating based on the tissues that their body produces. So it's just you are what you eat. That's really what it comes down to. We can measure those differences because even though it's only 1 or 2 neutrons worth of extra mass, at the atomic scale that has an impact, that has an effect. It changes subtly the way that chemistry happens because it takes more energy to move a larger atom, even if it's only heavier by 1 neutron. So the example here we have is carbon. Carbon typically weighs 12. It has 6 protons, 6 neutrons. Each of those contributes 1 atomic mass unit. Every once in a while, carbon will end up with another neutron. So because it still has 6 protons, it's still carbon. It still chemically behaves like a carbon atom, but it's a little heavier. And if you add 2 more neutrons, again, it is still carbon, but now you've added so much weight to the nucleus that it becomes unstable. It starts to radioactively decay. So when you hear about carbon dating, they're talking about carbon-14 dating because it decays over time, whereas carbon-13 is stable. It's the stable isotope in this analysis. And so whatever is in the fossil when it dies is likely to be the same ratio of heavy carbon to light carbon after the paleontologists find it and do the analysis. So the way we analyze it is using a stable isotope mass spectrometer. And don't get intimidated by the complexity of this diagram. It's actually relatively simple. You turn your sample into a gas, typically by dissolving it in some acid. So in the sample here we're talking about is like some powdered bone from a ground sloth. So you dissolve it into phosphoric acid, it bubbles up into a gas. You superheat that gas. You get the gas really, really hot and turns it into plasma, which is the 4th state of matter. So it's stripped of all of its electrons. And therefore, when it's stripped of all its electrons, it has a positive charge. So you can then fire it down this flight tube. And there's this giant, or not giant, but there's a big magnet that the ion beam hits. And because it's charged, it now is sensitive to electromagnetic fields. And so the beam turns as influenced by the magnet. And the lighter atoms turn more because it's the same force being applied to them than the heavier atoms. So the analogy I use is it's like if you're driving a sports car and you take a bend around a turn, you can take that bend much sharper than a large pickup truck that has to go kind of wide on that same curve because it's just got more mass if you're going the same speed, right? So when they get deflected, we now have these collecting cups, right? And the cups are placed exactly where we know the ions of different mass will hit. And the cups literally just count how many times they've been hit. Right? And so based on how many times they get hit with the different weighted atoms, you then have a ratio of how many heavy atoms versus how many light atoms you had in your sample. So it really works kind of like an atomic skee-ball machine, right? So you're just taking these atoms and you're rolling them up the hill. The hill in this case is just a big magnet, and then they fall into the right cup and you get your data. So how do we use this to determine diet? I talked about this a little bit already, but we know, based on prior research, that certain types of food, so in this case, the photosynthetic pathway that the plant uses, there's a couple different ones that have evolved, affects the carbon isotope breakdown, the carbon isotope ratio in that food. So we know if they're eating something that uses a C3 pathway, it's going to fall within one range of values. Something that uses a CAM pathway, like a cactus or succulents, has a different range of values, and something C4, like grasses and things like that, have a different ratio. And carbon and nitrogen are the only 2 I'm really going to be talking about today, but there's a bunch of other stable isotopes you can measure if you know what you're doing. Nitrogen tends to tell us about the trophic level, so where on the food chain is this organism. So primary producers, plants, tend to have low nitrogen values because they're getting it mostly from bacteria that fix nitrogen directly from the atmosphere, which is most of what our atmosphere is. Herbivores have slightly enriched nitrogen relative to the plants that they're eating, and then carnivores have the highest nitrogen values of anything because they're getting it from the herbivores that they're eating. So again, we mentioned harpy eagle being one of the few predators of the sloth. It's also the, I guess there's now a T-Rex skull, but before this was the only dinosaur in my talk. So it's a really useful tool. I'm going to cruise through these pretty quickly, but if folks have questions, we can get into it later. So you can use it to tell, this is data from manatees, which you mentioned before. You can use this to tell if manatees are eating a marine seagrass diet, a freshwater diet, or a freshwater C3 diet. You can then apply that to the fossil record. You can use it to determine trophic levels, so you can see that we have all these herbivores on the environment with relatively low values, and then all these carnivores with relatively high values. The anomalous outlier on that chart is the hippo because they're eating in water instead of on the land, so they just end up with a different value to start with. You can use it to determine dietary preferences within an ecosystem. So this is data from Yellowstone wolves and trying to figure out what is the preferred prey of Yellowstone wolves. And the way this kind of works is when you go to the paint store and you ask them to mix a specific paint, you're saying, put this amount of this color, this amount of this color, this amount of this color, because I want the final color to be this, right? So this process here is doing that in reverse. The wolf is our mixed can of paint, and we measure the wolf, and we know what the values are for the 3 main prey items, which are white-tailed deer, elk, and bison, and then we have a computer algorithm that essentially reverse calculates what different pigments could have been put into the paint can to get the final product of the wolf. So it's a really cool way of estimating what animals are preferring in their environment. In this case, you can see there's a very strong preference for elk over either deer or bison, because elk are probably in sort of the sweet spot of they provide a lot of food but aren't so big that it's a real challenge to bring them down. So I've outlined this really amazing technique. Why not just use that and call it a day? Because this is that question mark that we had from before. We don't know what happens inside the sloth when it eats the food. So we can measure the food that's going in, and we can measure the tissues that are coming out, like hair, claw material, feces, but we don't know how the values of the food changes as it works its way through the sloth's metabolism, especially since the sloth metabolism is so weird. So we can put together a pretty simple equation. We have our value of the tissue, our value of the food, and we need to know the offset is the term we use, and that's what these question marks are representing, is that offset value. So that was what I set out to calculate as part of my PhD research. These are my more scientifically formulated questions, and it's, do sloths have a different offset than most mammals? They're an outlier in so many other areas, is this one of them? Is a small amount of animal protein measurable in sloth tissues? So can we tell if sloths are eating meat? What does that say about our hypotheses for carnivory in fossil sloths? And then I'm not going to get into this one today, but what happens when you apply a novel offset value to existing fossil data? If you want to talk about that more, all the slides are just hidden so I can pull them up for folks, but we just don't have time. So I go to Costa Rica. This is the sloth sanctuary in Costa Rica. This is their mascota Buttercup and their greeter Pedro the Megatherium. And they have they're a rescue sanctuary, so injured sloths get brought to them, and I'm sure we have some folks here going into veterinary medicine, but the sad reality is sometimes when you get brought an injured sloth, it doesn't make it, and also some of the sloths that can't be returned to the wild just live out the rest of their lives at the sanctuary, so it is a place where sloths get a lot of treatment and good conservation work, but it is also a place where sloths die, useful for me, the paleontologist, need a lot of, so they have a large freezer, and so I go for a visit, and I go into the freezer, and I grab a trash bag full of sloth, and I get to work. So, it gets grosser. So I sample from as many sloths as I can at the time that I'm down there. It is not pleasant work. It is not my favorite thing to do, but it's important, and I think it's worth doing it on an animal that's already deceased rather than even stressing out an animal that's still alive by trying to collect tissues from it that it is still using. So I collected tissues from both genera, and I collected tissues from sloths that were categorized as 5-minute warning. Okay, I collected tissues from sloths that were considered captive, that they'd lived at the sanctuary for a little while before dying, and wild ones that had died almost as soon as they got to the sanctuary. And there's a reason for that. And I mentioned that it gets grosser. This tool here is called an increment borer. It's used in forestry to take cores out of trees so you can count the rings without having to cut the tree down. My PhD advisor, Mark, one of the people on my committee was in the botany department, and he had one of these in his lab. And my PhD advisor said, here's an idea. What if instead of thawing the sloth out entirely, you just use that to get a stomach sample by just coring in and pulling out a core of the stomach sample? So it turns out when the stomach is 30% of your body mass and it's all plants, that doesn't thaw very quickly. So it actually stays very hard. So it really, this didn't work. And the one time it came close to working, I was standing over the sloth trying to get the drill bit to bite into the stomach and actually get me some core out. And it finally pierces the stomach. It didn't fully pop, but this is hollow, right, to take the core out. And this is where my face is. And so I just got hit with a stream of fermentation gas, for lack of a better, yeah. I about lost my lunch. I was out, I had to leave, like I was running outside the room to get fresh air. It was so bad. So, yeah, so sometimes your advisor has a good idea, sometimes not so much. But I tried. So I'm gonna run through the results relatively quickly here, but you can see these are data representing the nitrogen values on the y-axis and the carbon values on the x-axis, and it's showing the hairs or the tissues that grow continuously, so things like hair and claw in all of our wild sloths, and you can see that the difference between the one portion of the hair and the other, it's all over the place. It's not going in any consistent direction. But when we look at our captive sloths, the captive sloths are nicely nested. So that suggests that, again, the wild sloths are having a more diverse diet. They're eating more different things, whereas the captive sloths are only being fed the diet that they're given at the sanctuary. It's good that we can see that in the data. And then our captive sloths, we can see that the values of their stomach and feces start near the tree that they're getting the leaves from and shift upward in a consistent way by about in a consistent direction and a consistent magnitude. So that's also really good. It suggests that we're getting a consistent offset in our data. So we now have some numbers we can fill in. You don't need to worry about these numbers but just see that we now have an answer to that equation we showed earlier. And there's a lot going on in this chart, but the thing to note is it's showing the different tissues with all the carbon ones on the left and all the nitrogen ones on the right, and it's showing how sloths compare to other mammals. And you can see by and large, there are times where they overlap with other mammals, but mostly they're falling outside the range of mammals that we know of that have had this technique applied to them before. So it is suggesting that the sloths are doing something different. Now to get through the idea of meat eating, so now we've got some numbers that we can apply to this question. We have a secret ace in the hole. And it's really strange that it worked out this way, but it gave me an opportunity to test the question, can we tell if sloths were eating meat? And it's because in the wild, we're pretty sure that sloths only eat leaves. But again, there are people that claim that they've seen sloths eating like eggs and small vertebrates and insects from time to time. So since about the 1970s, it's been standard practice with captive sloths to give them dog food. So what does that mean? It means we've got an herbivorous population of sloths, and we've got an omnivorous population of sloths. So we can test between the two to see if it's obvious that these sloths in the sanctuary are actually omnivores, right? So I won't get into the details too much here, but essentially you can look at the space between the two. There's two components of our bone, carbonate and collagen. They both contain carbon, and so you can measure the stable carbon ratios of both those elements, and what we found is that the space between, the delta between those two values tells you pretty definitively if that animal was consuming other animals or if it was an herbivore. And so a very simplified way of is, if your food source is 100% plant matter, your spacing on this big delta carbon chart is usually between 6 and 8. I did my PhD in Wyoming, hence the moose and puma and other things. If your diet is a 50-50 mix, so something like a bear, I think there's supposed to be a bear symbol here, then you fall between about 4 and 6.5. If you eat 100% animal matter, like this puma, then you fall between kind of 3 and 5. So our prediction here is we've got sloths that should plot as strict herbivores, same as the moose, and then captive sloths, which should plot as an omnivore, probably closer to the herbivore side of the scale. It's a low meat omnivore, which is a categorization we can use. So this has been done in the fossil record before, so this is another study that was done to try to answer this do sloths eat meat question, and you can see pretty plainly, this is a modern ecosystem on the left and a fossil ecosystem on the right. You can see that the wolves and the saber-toothed cats are plotting where we would expect a carnivore to plot, and our extinct sloths. And then I want to end my lecture with one brief update. on sloth paleontology right here in West Virginia. So we are in Pocahontas County at the moment. I put a star roughly where we are. I'll get to this in a second. Last year in Putnam County, something amazing was found. So this is Kevin Adkins. He is a turkey hunter and works in construction, but works in construction in a way where he like knows the land really well, knows geology pretty well too. He went out turkey hunting on Mother's Day last year, and he and his father-in-law were together and hiking up this creek in Putnam County, and they saw this. It had just been a heavy rain, so a hill slope had kind of washed out from this creek and a bend in the creek, and they saw this thing caked in mud that they thought they could see some bones and some teeth sticking out of. They thought it might be a big cow skull. So they didn't end up bagging any turkeys that day, but something I learned is apparently when you say bagging a turkey, it's because you literally, when you go turkey hunting, you carry a thing called a turkey bag with you. It's a big bag to put your turkey in if you kill one. So he had an empty turkey bag, so he found this thing and just brought it home with him. And Stark, you were asking me about the size before. I mean, that's how big it is. It's a large animal. So this is a almost perfectly preserved Megalonyx jeffersoni, which is the state fossil of West Virginia. And I showed you the fossils at the beginning of the lecture with just a couple of hand bones. To my knowledge, no skull of a Megalonyx has ever been found in West Virginia until now. This is an amazing discovery. I took some 3D scans of it when I got to see the fossil in person, so we can just kind of see the cool 3D scans. So you can see, I mean, it is so perfectly preserved. This part of the bone right here is called the zygomatic arch. That never lasts. That always gets broken off. There's small little bones inside our nose called nasal turbinates. This still has those intact. It's incredible. It's lost a few teeth, but other than that, it is an amazing, amazing specimen. So why is this such a big deal that we found this where we did? So again, this is roughly where the skull was found in Putnam County, West Virginia. The red dots on this chart over here in the eastern part of the state represent all the known caves. And the cave from the original sloth fossil is one of these ones down here. So this is a limestone region of the state. Karst topography is what it's known as. And it's got a lot of caves, and so we find a lot of fossils in caves. There are no caves in this region of the state. It's a completely different geology. And this fossil didn't come out of a cave. It washed out of a muddy hill slope and was like caked in clay and mud. So all the sloth fossils that we have, including the ones in Virginia and including the ones in Maryland, come from this strip of limestone caves. And we've never found one from this part of the state. So how did it get there? Good question, right? It walked, yeah. When it was alive, yes, but how did it get preserved there? So we don't know yet, and this specimen is still being held by the family that owns the land, which is not Kevin's family, and they haven't yet donated it to science. So there's not much we can do on this specimen until it gets donated to somewhere we can actually appropriately do science to it. But the working theory we have right now is that during one of the previous ice ages, a glacier came down into Ohio, didn't make it all the way to West Virginia, but it dammed one of the rivers that flows into what is now the Ohio River and created this giant lake. So this is a lake about the size of Lake Erie today that existed in a theoretical lake that existed in southwest West Virginia when this glacier was in place, forming a glacial dam. And the sloth fossil comes from right here. And the elevation it was found at, in the modern elevation it was found at, is about the elevation we think was the lake's shoreline. So I think this sloth slipped and fell in a lake and either drowned or died and got covered up in the mud. And all these little brown splotches on this chart that was done using GIS, we think were islands. We think these are hills that were tall enough that they would have been little islands. So I think we might have found an island hopping sloth that unfortunately met its demise, but got covered up in this really soft lake sediment and preserved until a turkey hunter found it last year. So some conclusions to think about. We're still figuring out, the question of, is the chemistry going on inside modern sloths a better approximation of the chemistry inside ground sloths than any other animal? It does provide interesting results to think about. No evidence of ground sloth cannibalism, and we do get something different when we use sloth-specific offsets. And so I use this image at the end of my talks because one thing I like to say is that this type of science I do is I'm trying to use the past to inform the present to inform the past. So I'm taking paleontological techniques, applying them to modern animals, and then seeing how that data informs our interpretation of the fossils. So it's sort of an Ouroboros of the snake eating its tail of research to try to use the past to inform the present to inform the past. Big picture take-home messages. Buttercup did die in 2019. She actually died the day after my lecture here at NYSCamp. So was yeah. I like gave this lecture, and then Stark remembers I was pretty broken up about it. Yes. No, Buttercup is too important a mascot and an ambassador, quite frankly. She was raised at the sanctuary her entire life. Her mom was hit by a car, and she was brought to the sanctuary as a suckling infant still. And Judy, the woman who runs the sanctuary, was able to keep her alive and nurse her back to health. And she's one of the few three-fingered sloths that has ever survived long-term in captivity. And so the age she died at, I mean, she was 1992 to 2019, which is a really long time for a small-bodied animal. I mean, think about like dogs and cats and stuff. They don't live that long. And she probably would have lived longer if she hadn't had some medical complications. So sloths live for a really long time too, which is weird. So sloths are really weird. Hopefully I've emphasized that enough throughout this talk. And then I think it's really interesting that the idea of this fossil being found in Putnam County, paleontology may be about what happened a really long time ago, but there's still new and exciting stuff being discovered and found out all the time. So just something to keep in mind. And for all of you to think about as you go through science camp and go through your college careers, if you pursue a research project of your own, you'll end up with answers, and you'll probably end up with more questions than you even have answers for. And so almost every research project I've had ends in something of an answer, but usually like 3 more branching questions off that answer. And so you really can like go on kind of forever if you are curious and just want to keep answering questions that come up as you try to answer the first question you had. So with that, I want to thank everybody who helped make this research possible. I did not get here alone. Friends, family, professional mentors, and folks like the folks who work for the foundation and the academy. So I appreciate everyone who's helped me get here along the way, and I appreciate all of you for listening. Thank you so much.
Awesome. Thank you, Ryan. We don't have time for questions, but good ones along the way. So we have programming that is going to happen. Please do not go back to your bunks. Do not go anywhere. But if you need to go to the bathhouses and then straight to the benches by the flagpole, we'll be doing a tour of camp. Also, unless it's absolutely necessary, please use the restroom before or after lecture.